Welcome. In this lecture, I will describe viral reproduction and classification. The first viruses to be studied intensively were the bacteriophages, or viruses which infect bacteria. These types of viruses are easy to culture and study because they can be grown on lawns of plated bacteria. Bacteriophages can be classified into two main groups based on their life cycles. The first are the lytic phages. Their name comes from the fact that these viruses always lyse or pop the host bacterial cells. These destructive phages leave behind plaques on bacterial cell lawns. The series of figures on the right outline the key steps in lytic phage infections. First, the phage must attach to the bacterial cell wall. Next, the phage DNA is injected into the host cell through the cell wall. From this position, viral replication, transcription, and translation of viral proteins occurs. Hundreds of new viral particles are assembled within the bacterial cell, which eventually pops or lyses, releasing the new phages to infect other cells. During the course of an infection, the viral DNA is kept separate from the bacterial host chromosome. Lytic phages have been extensively studied for their usefulness as an alternative bacterial infection-fighting strategy. Unfortunately, phage therapy has not been perfected yet. Maybe it will be in your lifetime. The second group of bacteriophages is called the temperate or lysogenic phages. This group of viruses is less destructive because they don't always lyse their host cells. During the course of the infection, the viral DNA becomes integrated into the host cell chromosome. Viral DNA, shown here as pink, becomes integrated into the host cell chromosome. In this position, it's known as a prophage. The bacterial chromosome is seen as purple. Depending on environmental conditions, the viral DNA will be replicated along with the host cell for many, many generations. At some point in the infection, these viruses may become lytic, replicating inside of their host cells and eventually lysing them. The lysogenic phage cycle is important because it allows a process called transduction to occur. Transduction is the process of bacterial DNA being transferred to other bacteria through a phage infection. This is an important mechanism for the spread of antibiotic resistance. The textbook website has some excellent animations to illustrate this process. Check them out. In addition to transduction, lysogenic phages are interesting because their life cycles are similar to latent infections, which are seen in some clinically significant animal viruses. Latent infections occur when certain animal viruses integrate their DNA into human host chromosomes. These infections are not cleared by the immune system and may persist for the lifetime of the individual. In some cases, the infection is subclinical or without further symptoms after the initial acute infection. In other cases, the patient will experience recurrences which, depending on the individual's immune system's ability to keep the virus in check. A latent state is said to occur when viral DNA is found within the host chromosome in a proviral condition. This table lists a number of common viruses who become latent in their human hosts. It shows their primary acute disease and the recurrent disease, as well as the cells which carry the viral DNA. For example, the varicella zoster virus causes primarily chickenpox, which can later occur as shingles. The cells which carry the viral DNA are found in the dorsal root, which are sensory ganglia along the spinal cord. I will discuss this virus and other latent viruses in more detail later in the course. Animal viruses are an extremely diverse group of organisms. They can be grouped together based on their genetic information, or genome, the presence or absence of an envelope, their shape or morphology, the type of cells they infect, or the type of diseases they cause. The most current scheme of viral classification is called the Baltimore classification system. This system groups viruses into 7 viral classes, depending on their genome type, how they replicate, and how they produce viral messenger RNA. For example, the herpes viruses contain double-stranded DNA, which they use to make their messenger RNA. Herpes viruses are classified into Class 1 in the Baltimore system. In contrast, retroviruses like HIV contain single-stranded RNA, which they must first reverse transcribe into DNA, which then they use to make messenger RNA. HIV is classified into Group 6 by the Baltimore system. Viral family names end in viridae, while viral genus names end in virus. Let's look at some important examples on the next 2 slides. Table 13.2 contains a summary of the major viral families that affect humans. Bio 15 students are required to memorize only some of the information contained here. For the starred viral families, students need to know the family name, whether the virus contains DNA or RNA, and the major disease or diseases caused by members of that family. For example, the Papoviridae are DNA viruses which include the human papillomavirus, or HPV. HPV is usually sexually transmitted, and it is associated with certain cervical and other cancers. Students don't need to know the size of the virus. They don't need to know whether it's enveloped or non-enveloped, whether it's single-stranded or double-stranded. Other viral families students need to know include the Poxviridae, which are viruses which include the eradicated smallpox virus. The Herpesviridae is a large DNA family which includes the herpes simplex viruses and the chickenpox virus. The Hypnatoviridae is another DNA virus family which include the causal agent for hepatitis B. As for RNA viruses, they're seen below. The first RNA virus is the Picornaviridae, which include the polio and many cold viruses, and the causal agent for hepatitis A. The Calciviridae are RNA viruses which include noroviruses, which cause the 24-hour stomach flu. The Togaviridae, seen on the bottom, are RNA viruses, which include the causal agents for mosquito-borne encephalitis and rubella. The Flaviviridae are RNA viruses, which include the causal agent of dengue fever, the hepatitis C virus, and West Nile viruses. Coronaviridae are RNA viruses which are associated with respiratory infections, from SARS to the common cold. Rhabdoviridae are RNA viruses which include the rabies virus. Orthomyxoviridae are very important RNA viruses which include the influenza or respiratory flu viruses. Finally, the Retroviridae are unusual RNA viruses which use reverse transcriptase to turn their RNA into DNA. Retroviridae include the HIV virus, or the cause of AIDS. All of these viruses and the diseases will be described in more detail later this semester during the diseases portion of the course. This concludes Virology Part 2.